Hello and welcome to CureTalks. This is University of Pennsylvania's COVID Talks series, where we discuss latest trials and studies on COVID that Penn is part of. I'm Priya Menon, and with me today is patient advocate Cynthia Schmilowiski. We are talking about the Convalescent Plasma study with the Principal Investigator, Dr. Katharine Barr. Dr. Barr, welcome to CureTalks, and thank you for being here today.
Thank you for having me.
Dr. Barr, let's just start with a very simple question. Can you describe the theory behind the convalescent plasma and what we are trying to achieve with this trial?
Yeah, so good place to start. So the theory behind convalescent plasma, or the biological plausibility, is actually quite old. For many infectious diseases, for a long period of time, people have used plasma as the carrier of antibodies as a treatment. So for infections as old as the flu and other types of viruses, before we had specific targets or antiviral medications, we had been thinking that people who were previously infected and made an effective immune response, we could take plasma from those individuals, take the hopefully effective and well-formed antibodies from those individuals and give them to another person who was suffering from an acute disease. So this has actually been going on for a long time. There are data in other infections. They're not overwhelmingly positive, but the convalescent plasma has been used and shown some benefit. And then the next step of that process for other infections is that people will take out the antibodies, either making monoclonal antibodies or other antibody formulations, and that is considered standard of care for several different infections. So for COVID-19, we're kind of in the same situation. We have a new virus where we haven't had enough time to study it and develop a full range of specific antiviral drugs, nor pull out the best kinds of antibodies and make antibody-based therapies. And so we're going to the primary source, and that is people who have recovered from COVID and have detectable antibodies in their plasma, and using those plasmas to treat acutely infected individuals. So that's the basic premise of the trial. So here at Penn, the blood bank, led by Dr. Don Siegel, Nicole Acquis, and Andrew Femster, have been collecting plasma from individuals who had COVID disease, tested positive during their acute infection, recovered, and then were at least 28 days, a full month of symptom-free days. Afterwards, they went in and donated blood. And so from those individuals, we've collected plasma. We've tested that plasma with an antibody test to make sure that there are at least some level of antibodies against COVID. And then those people's plasma has been stored in the blood bank. And then, so that's one component of the operation. The clinical trials that we are running for patients in the hospital, there are two of them, but the basic premise is we take people who are acutely ill and sick in the hospital with COVID, and we give them plasma from one or two of these donors who have donated convalescent plasma. The plasma is matched to their blood type. And again, it's just been screened to show it has at least some level of antibodies. And we give that to people in the hospital, and we monitor them to see if it helps. Now, there have been a couple reports to date of this sort of convalescent plasma strategy. So there are several small trials out of China where they gave convalescent plasma to somewhere between 5 and 10 patients each. And these are not randomized controlled trials. They're just more observational of what happened. And what those trials suggested was that it was safe. There weren't a lot of bad reactions to the plasma, and at least some of those people seem to get better, either have their breathing improved and potentially leave the hospital. There is a large study that was recently published by the Mayo Clinic for an expanded access program where they looked at thousands of individuals around the United States who received convalescent plasma, and they don't have data about how well the convalescent plasma affected their disease course, but it was shown to be safe with a very low level of of adverse events or bad reactions or bad clinical events that happened around the plasma. So where we are at Penn, and the goal of these trials is to test this a little more rigorously in a scientific manner. So we have two trials. The first is looking at our sickest patients, those who are intubated and mechanically ventilated, who can't breathe on their own. And so we're going to follow those individuals after they get 2 units of plasma and ask whether this plasma improves their condition. We're going to compare the results clinically to a different trial conducted by the NIH, in which one arm were placebo recipients. So they basically were similarly sick with COVID, but didn't receive any intervention. So it's not a perfect randomized control trial, but at least it's a group of matched participants that we can compare and say, how did the clinical outcomes differ in this group that got plasma versus this similar group who did not get plasma? Our second trial is for individuals who are a little less sick. So you have to be hospitalized and have pneumonia and have difficulty breathing with your COVID, but you don't have to be in the intensive care unit on a mechanical ventilator. And we are that's an 80-person trial where half of the people will get plasma and half won't. And we'll compare, does standard of care plus plasma, the outcomes, are they any different than standard of care alone? And so hopefully with these trials, we can take this atmosphere where we have optimism and we think that plasma could help, but we can have a little bit more rigorous understanding of whether it actually is safe and efficacious for patients who are quite sick in the hospital with COVID.
That's very interesting. I know my next question was actually on the eligibility as to who can donate plasma, but I think that you've already answered, saying that somebody who's recovered from COVID-19 is eligible. Is that right? Yes.
Yeah. So right now for this protocol, we are required to have people who had an actual diagnostic test. So while they were sick, they went and got one of the nasal swabs and were shown to have the virus. So if you had that while you were ill and then you've recovered, then you are eligible to donate plasma for this study. If you just suspected that let's say you had a contact who was diagnosed and you had very similar symptoms, or you had a disease course that sounds a lot like COVID, but you didn't have the opportunity to get tested, there are still clinical trials here at Penn that you can volunteer for, and you can contribute to the scientific effort in great ways. But per the FDA guidance right now, we are not allowed to use those individuals' plasma as a part of this therapeutic program.
Okay, so I like a little bit more detail about as to how this plasma donation is done. Like, is it matched? Like you match blood, for example? Would it
Yes.
be plasma matched with patients? Yeah.
Yes, exactly. So we use compatible blood types. So it's similar to blood cell products, except what we have in the plasma is the antibody reaction to whatever types of cells you have. So basically we have to make sure whatever blood people receive, the antibodies in the donor plasma aren't going to react to their cells. So it's similar. If I'm A type blood, I can receive A type plasma. O type blood can receive O type plasma. It's similar. It's a similar concept.
Oh, that's clear. With that, I'm going to hand over to Cindy for her questions. Cindy?
Thank you. Thank you, Dr. Bark, for explaining lots of this. I want to talk a little bit more about the target population. You explained that there's going to be 2 trials going on, one for the severely ill and one less severe. In both of those populations, if you choose to have the convalescent plasma, does that exclude you from any other of the treatments, like with remdesivir or any of the other promising therapies? Or
that's a really good question, because we're kind of in this gray zone right now where there's lots of new and evolving, maybe promising therapies, but we don't know which one is better. So it's hard for patients and patients' families to decide which trials they should participate in. Because this is a phase 1 study and we're really trying to determine what the safety and the efficacy are, we can't enroll a person who's getting this therapeutic trial, getting this experimental therapy in another phase 1 study. So a patient who agrees that this convalescent plasma is something they'd like to participate in, they are excluded from most other COVID therapeutic trials. But that doesn't mean they can't get a lot of the other experimental therapies. So for instance, remdesivir is now available via an expanded use program where the government is providing certain hospitals with a certain amount of the drug. So if that drug is available, and it's honestly it's not a sure thing how much of that drug is going to be available for which patients or for which hospitals, but if the drug is available, a patient who's getting plasma is still able to receive that drug. If there's a different trial that is not about COVID, then that would be fine. But in order for us to fully distinguish what the safety signal is from this plasma, we can't have multiple experimental trials at the same time. Otherwise, we won't be able to tell if something happened. Is that because of the plasma, or is that because of the other experimental therapy that we're giving you?
Okay, but you're still able to give other therapies that are not experimental.
Right. So, well, yeah, I mean, so that's kind of the gray zone because everything is experimental for the most part. We have a little data about remdesivir. So remdesivir is now considered sort of one of the standard of care medicines. We have some data about hydroxychloroquine, et cetera. And so some people have been giving that as a part of routine clinical care. Anything that your medical team thinks is appropriate for your medical care can be used. It's just the experimental clinical trials that you're limited from.
Okay, and a little bit more about the target population and the eligibility criteria. Are any groups of people excluded from this trial?
Yeah, we're trying to make this a pretty broad trial. So for our first trial, it's called Penn CCP1 or Penn COVID-19 Convalescent Plasma 1. This is for our sickest patients, our critically ill patients who have mechanical ventilation. So the only exclusion criteria are that you need to be on a ventilator, you need to have a recent test showing that there's somewhere in your respiratory tract there's virus. So we want to make sure that we're actually treating COVID and not another issue. We need to make sure we have the right blood type available for the plasma. And then we want the study team and the medical team to think that COVID-19 is actually driving a major part of the disease process. There are a lot of very sick patients with COVID who maybe get over the COVID, but they're dealing with all the other problems that happen from very sick patients in the hospital. So we don't want to give plasma to an individual where we don't think it has any chance of helping. So we're trying to give it to people who we think the active virus replication is causing the problem. So those are really the exclusion criteria. But for instance, if your kidneys aren't working well or if your liver is inflamed, those are all fine.
And is this a trial that you have to be offered, or could someone ask to be part of the trial?
Yeah, so what I should mention, sorry, the second trial, it's very similar criteria, except you don't have to be on a ventilator. You just have to be hospitalized, have evidence of pneumonia, and have the COVID test be positive, and have your team think that COVID is driving disease. Yeah, so these studies are only currently being offered at our Penn hospitals, so at the Hospital of the University of Pennsylvania and at Penn Presbyterian, because that's where our blood bank has a relationship. So we look for all eligible patients within the hospital system and try to enroll those individuals. If a specific patient or a patient family has an interest in plasma, that at times they've brought it to our attention that the family is interested, but we are trying to sort of get as many people who we think would be good candidates enrolled as possible. So if people were to request it and be hospitalized in our system, that would be great.
Now let's talk a little bit about the plasma donor. What's the donation process? Is it just a blood draw, or is it apheresis? I know when I got my stem cell transplant, I had to be on an apheresis machine to get my plasma cells out. So what about the donation process?
It's similar to what you went through. It's not exactly the same process, but it's the same sort of machine. So what we do with the plasma donation process is you have a couple of large IVs placed in your arm, and instead of instead of taking the whole blood donation, the blood gets processed through the machine and your cells get returned to you, and they just take some of the plasma. So it's, like you said, it's an apheresis machine, and they're taking the plasma in a plasmapheresis process and giving you back your cells, your red cells, etc. So it doesn't make people quite as anemic as a regular blood donation would.
And how long is that process usually?
I think it's about 1 or 2 hours.
Okay, and do they get enough donations for several patients, or is it a one-to-one donation?
That's a good question. So what we're doing right now, I think that the plasma team has gotten about 50 or 60 donors so far. And each donor gives about 2 units if they can, if you can get that amount. Each unit is about a cup of blood, so that's about 2 cups of plasma. And we actually end up giving each recipient 2 cups of blood, but we oftentimes don't give the same one person's 2 units to the same recipient. So in order to try to mix and match some of the antibody levels, we've tried, if we can, because we have flexibility with the blood type, to give plasma from 2 donors to each recipient. Sometimes for the more rare blood types, we aren't able to do that, but that's been our goal.
So the recipient gets 2 doses of the plasma at the same time, or is there a time between it?
Yeah, so we give each unit of plasma goes in over about 30 minutes to an hour, and then we break for an hour to monitor and make sure there's no infusion reactions or issues, and then we give the second unit. So it's within the first few hours. If the participant, if it has any reasons that they need to slow the infusion, they're welcome to do that, but most people tolerate the infusion within just a few hours.
Okay, great. And I know you had said earlier that the donor had to have a positive COVID-19 test. Do you test for antibodies also prior to the donation? Is it just yes, no, or is there like titers testing that they have to have a certain level of antibodies?
That's such a good question. I think that's really the heart of the matter. So there's 2 things. So for clinical trials, we have to use FDA approved sort of a certain high level of clinical grade assay in order to make any clinical decisions. So because we're relatively early in this disease process, we don't have a lot of great antibody tests that are validated for this sort of thing. And we don't actually know what the type of antibodies or the amount of antibodies is that's needed to be an effective therapy. So for this trial, all we are doing is a qualitative test where we show by an ELISA assay that there are COVID antibodies. So it's more of a yes or no. What we're going to do on the back end of this test is we're going to follow those antibody levels both in the donor before we give the donor plasma to the recipients, and the recipient at baseline before they get any plasma, and then over time after the recipient gets the plasma, so we have a good sense of, in a much more sort of thorough way, what the quality and the quantity of those antibodies are, and we'll be able to correlate that a little bit with the clinical outcome. And so that's more sort of translational research level information, but it will hopefully help us understand in this therapeutic scenario what types or amounts of antibody are needed to improve. What types of patients? If you have a patient who's been sick for a long time and they actually have high levels of antibodies, could they be helped by an antibody transfusion or not? If you have someone with very low level antibodies, are they more likely to be helped by a certain type of antibody? So there's lots of questions that will give us answers about whether convalescent plasma is effective, but also help us move into the next line of therapy where people make monoclonal antibodies, or maybe a mixture of antibodies that could be used in a more directed way for the next round of therapy against COVID.
Okay. That's why I was kind of looking into the future there. So
yeah, that's a great question. You hit the nail on the head. That's what we all would really like to. We would love to be able to just choose the most effective high titer antibodies and give those directly to these patients. We just don't know yet what those are. And it's kind of a limited resource because there aren't that many people who were diagnosed with COVID and are now recovered who are coming in to donate. So we're having a good response and there's lots of people, but there aren't an infinite number of people who had COVID, had the test, and are now feeling great and want to come in and donate.
And they have to come to your facility? Is that where
so they can come to the Penn blood bank. And if anyone's interested, if you Google search the Penn, Penn COVID volunteers or plasma, there's a donor registry that you can sign up to donate plasma if you're interested, or volunteer for various other studies.
Can you donate more than once?
I believe so, but I'm not, I believe you can. up to twice, I believe, for the plasma antibodies. But all that information would be available on the website as well.
Okay, and how long can that bag of plasma be kept in? Is it like frozen in liquid nitrogen?
Yeah.
Or how's that?
Yeah, so when people donate, their plasma is frozen, and it can be kept for quite a while when it's frozen. And then we don't thaw it until we have a matched donor that we think is appropriate. And then once that bag of plasma is thawed, it can actually be kept in refrigeration for a couple days. But we generally use it once we thaw it.
Okay, great. And besides, I guess, on your informed consent form, there's always the risk and benefits.
Mm-hmm.
Besides not working, what other risks do you think are associated with receiving the convalescent plasma?
Yeah, that's a good question. So fortunately, plasma is given all the time during routine medical care, so it's actually a fairly safe clinical procedure. We do it for lots of different situations, most often if people have bleeding problems or if they've lost a lot of blood, but we have a good understanding of how safe this is. So the specific risks are that people could have an allergic reaction. If you've ever had an allergic reaction to blood products before, you're more at risk for that, so we would make sure we asked about that and determined your risk for that. It's only 2 cups, but actually 2 cups of volume is potentially a lot of volume for some very sick patients. So there's the possibility of sort of a volume overload situation that the clinical team would need to monitor and either, encourage urination or dialysis or something to keep the volume status even. And then there's very low level or sort of theoretical risks that we don't know if they're real or not, but given COVID's, the level of uncertainty we have about COVID that we usually mention. So we know that with COVID, there's an increased risk of blood clots in the leg, blood clots in the lung, even strokes and heart attacks. And so most COVID patients, they're being very closely monitored for that, and they're anticoagulated to prevent that. Now, it's been shown if people get lots and lots and lots of blood products that you can increase their risk for clots. So in theory, plasma may contribute to that, but it's probably a nominal increase in the risk of clotting, and it's something we're already very aware of and trying to prevent in our COVID population anyway.
Good. In the future, right now it's kind of a one-to-one donation, like one bag, or now maybe we're getting plasma from 2 different people. But in the future, is there any idea of maybe making a pooled plasma donation, kind of like IVIG, where you might have a number of different people's antibodies in one infusion?
Yeah, and that's the way a lot of these antibody-related products have been delivered for other infections, is through an IVIG type of formulation. I think what will happen with COVID is there are a lot of groups right now pursuing monoclonal antibodies. So these are specific antibodies they've pulled out of people who had COVID, and they're basically searching for the ones that are the most potent neutralizers and are, the most effective. And I think these will be sort of developed as products, and there will either be a single monoclonal or a mixture of these monoclonals that could be used as a therapy. These same sort of things could be used as a preventive strategy also. So there's a huge amount of scientific effort into identifying and characterizing these monoclonals. And I think we're going to see phase 1 clinical trials in the not too distant future, testing these antibodies for both prevention as sort of a passive immunization, like a passive vaccine process, and as therapy. If it looks like these antibodies are effective in the sort of convalescent plasma type mode. So I think that's probably the next step.
Great.
And that takes away some of the issues with blood transfusion and volume and whatnot. It just directly gives the antibodies.
And one kind of last question, I guess, because you're on the line and I get to ask you this question. What if you had your crystal ball, and for people like me who have multiple myeloma, who have a hard time producing intact antibodies anyway, what would convalescent plasma look like for us? Would it be something we would want to consider? Or is that something that maybe I won't be able to make those antibodies?
Yeah, so for people with any sort of immunodeficiency where their immune responses are a little bit weakened, you might be a perfect person to consider a monoclonal antibody as a preventive therapy as opposed to just vaccination. So hopefully we'll develop a good vaccine that we can give to almost everybody. And that basically is a part of the virus that your own immune system responds to by making good antibodies and good T cell responses. But for some people who are maybe a little immunocompromised and aren't able to mount that good active vaccination response, this sort of passive vaccination concept where we just give you good antibodies through delivery of a long-lasting antibody, monoclonal antibody, that might be a good pathway to protect people like you who want to go. I mean, hopefully we're able to get out and walk around and do things in public again. And for people who may not be able to have the highest or the best vaccine responses, a monoclonal antibody product might be a good sort of surrogate for that.
That sounds promising to me. I would really like to get out of my house someday.
I bet.
Well, thank you so much.
No problem.
Maria, do you have any other questions?
No, Dr. Barr, thank you so much for your time. I think it was a real great pleasure to talk to you and learn about the study. And Cindy, thanks for joining today. Stay tuned to Penn's COVID Talks series to learn more about breakthrough COVID research happening at Penn. Thank you, everybody, and stay safe.